Welcome to the Wednesday Yachting Luncheon, live from the virtual grow room of the St. Francis Yacht Club. We hope that you and your family are safe and comfortable, sheltering in place in a thoughtful, protected environment. And we look forward to greeting you back at our yacht club, our beautiful yacht club, just as soon as conditions are safe enough for gatherings. We're asking a fascinating question at the Wednesday Yachting Luncheon today. How do the smallest organisms thrive in the biggest oceans? And why would we care? Well, we care for a very good reason. It turns out that on Earth, half of all the conversion of carbon into organic compounds occurs in the oceans, not on land at all, but in the oceans, with microscopic plankton doing most of the heavy lifting. These tiny workaholics are hard to study in the ocean's deep and vertically stratified environment, and thus the conventional approaches to sampling plankton until now have focused on large populations of these microorganisms and have typically lacked the resolution to measure individual behaviors and processes of individual tiny plankton. As a result, worldwide scientists know very little about the microscopic biological and molecular processes in the ocean, such as how these little teeny microscopic plankton sense and regulate their depth, and even more interestingly, how can they suspend in the ocean, and how can they change their depth up and down and regulate their height in the water column with no appendages of any kind. They don't have fins or flippers or anything. They're smaller than the head of a needle. How do they do all that? Well, to answer that question, we've asked two Stanford bioengineering researchers to tell us about their research. Manu Prakash sailed lasers in the Charles River, which is a fun thing that many of us have done, beautiful environment to do it in, and he's bombed around the bay in catamarans. And Deepak Krishnamurthy has been on ferry boat rides off of Mumbai. Some of us have done that as well. And he's raced around in research catamarans in Hawaii, a wonderful way to look at the environment. But when they look at the environment, they have different questions in their mind than you and I. They're not looking at the wind shifts so much as they're interested in what's going on inside these little microscopic organisms that are too small for any of us to see. So Manu and Deepak, welcome to the Wednesday Yachting Luncheon. Tell us about your fascinating research.
Thank you for having us.
Thanks, Ron. Thanks for having us.
One of the things that I wanted to bring up is we as scientists are both deeply excited about engaging with the sailing community and many of the connections you will see very briefly again, but also we are excited about engaging all of you in the scientific tools that we build and share to help us understand the mysteries of the ocean. And I think before I begin, I want to start with just a brief land of the lab. We are based out of Stanford. We've been here for the last 9 years. And much of the work that you'll hear is done in collaboration with students in my lab. And I think the joy of just working with individuals, and again, Deepak is right in that picture there. A few members that are not present here today, Hong Chuan Lee, Adam Larson, and then another sailor. Until the last 6 months ago, I had a sailor in my lab who was from Brittany in France, Thibaut Paulina, who is also part of the broader project that I'll talk a little bit about on the Plankton Planet side in the very end. And it's quite a joy to have a very interdisciplinary group of people in the lab, and many of the projects that you'll hear about today are the result of that interdisciplinary group itself. So today we'll talk about two specific ideas and technologies, but they're really both at the heart of plankton. And no better way to begin is with this little painting that an artist in the lab, Rebecca Conte, made. And for the next hour or so, we want you to think about what if the ocean had the chasm cut in deep inside, and you could literally see and look through that thin layer of a whale that we all often spend time in the ocean for. And this view is extremely important to understand and appreciate, which will become very clear, to see what lives in the ocean, and especially the microscopic life, how it lives and behaves. And no better way to begin is also, going back to the words of Rachel Carson herself. And I think or two to just read through, but the two lines that fall through in here are: the ocean is a place of paradoxes, and many of you might have appreciated this before. It is the land of the great white shark, the 2,000-pound killers of the sea, but also the land for small things, so small that your two hands might scoop up as many of them as there are stars in the Milky Way. And this has really been the heart of what we've been thinking about, is how this life at microscopic scale shapes much of how oceans actually work. And we're going to define that slowly. And this is one picture of a comparison that often enough we think of our oceans covering majority of the surface of the planet. But many of the properties that we associate with the ocean are linked very deeply to the microscopic life that we often don't even get a chance to see. And in the perspective, Deepak, I don't know if you want to walk through some of these slides, the next few sets of slides.
Yeah, I think as Manu mentioned, and as you saw in that quote, it's really it's a system with a lot of paradoxes, and it's the largest biosphere on our planet, but a lot of the really important processes actually happen at the microscopic scale. And like one very striking example of that is that the ocean plankton actually fix 50% of global carbon. And they're also responsible for half the oxygen that we breathe. So sometimes the way we think about it is that if you take 2 breaths, every other breath you have plankton to thank for. And so this image that you see on the screen is actually an example of like a model or a simulation of like phytoplankton concentrations in the ocean. And only now we are beginning to understand this really incredible and like extremely connected ecosystem that connects the whole planet and really determines atmospheric carbon levels, and therefore is a big player in climate.
So what we'll do today in the next half an hour is tag team along and tell you completely new ways of studying this life forms in the ocean. And before we do that, I think it's extremely important to also just understand in the changing climate that we are starting and have been watching this shift, at least between 30 to 50% of all the carbon that humans as a race have been emitting is actually being absorbed by the ocean. So oceans in some sense have acted as our lifesavers on this planet. But the processes, and especially the biological processes that are at play that enable this remarkable phenomena to occur have really not been studied in the minutest of details. And so one of the things we will talk about is, of course, the enormous beauty of life in the ocean, but keep in the back of your mind that there is an urgency associated with this, that if we don't understand this, we will fall into a trap where majority of the largest carbon sequestration technology that naturally occurs on our planet might be on the edge. So many of you have been in the oceans, so you can see the sense of the density of life sometimes. This is a video taken by David Hannan from Plankton Productions from a short dive off of Papua New Guinea. And this is really at the heart of the challenge to study life in the ocean. This is the level of density. And again, this is all centimeter scale organisms. The microscopic organisms are even in a higher density. And how do you really understand the complexity of the web of life that forms? And we're going to present 2 uniquely different approaches to the study. But remember, this is the kind of biomass that you might be dealing with when you're looking at the ocean. Again, going back to the simplest possible scenarios of the class of the food webs, to say, most of us think about what comes as products from the ocean as the larger fish and, of course, the larvae that produce. But in the end, there is a very important connection of the ocean to the atmosphere, which is the dissolved gases, both the oxygen that many of these plankton will produce and the carbon dioxide that they will absorb. And effectively, much of this food web, when many of these organisms die, they sink. And that sinking is essentially at the heart of the sequestration technology that we'll talk about. And then we have to deeply understand how life in the ocean actually sinks. So again, going back to the sets of simulations that Deepak just briefly referenced to, the heart of the challenge, there's twofold. One is, how do we understand these living forms in where they are in the vertical depths of the ocean? But the other challenge that exists to us is that the ocean is a very big place. How do we measure simultaneously in different parts of the ocean how this living system is breathing and changing over time? And this is also where sailors come into play, which we will talk about in the very end. So we will now jump to the first part of the technology, which we call the gravity machine. And no better way to actually see it in this form right here. At the back, you're seeing some of the, of course, the individuals involved in this project and the machine itself. And I'm going to jump into maybe talking about the vertical aspects of the ocean. So Deepak, if you want to walk through this.
Yeah, so I think in these simulations, like these global scale simulations that you saw, you're kind of looking at like surface concentrations of these phytoplankton. So the key thing that, some of us, we should really keep in mind when thinking about the ocean, and this may be something when you go sailing, this also becomes apparent through, many phenomena that you have to deal with when you have to sail, is that the ocean is actually extremely vertically stratified. And, because of gravity and the density of seawater and the way that light interacts with water, what happens is that most of the things that living organisms care about, like light concentration, salinity, temperature, all of that change much more rapidly as you go down in depth than if you moved in the horizontal plane. And I think like a very striking example of this is just pressure, where for every 100 meters in depth, the pressure actually increases by 1 atmosphere. And so what this means is that like these biological processes that the plankton are going through is happening in this extremely stratified vertical landscape. So the question is, how do you actually study something like that without actually dropping a microscope in the ocean?
And I think, speaking of dropping, I want all of you to think about this. I'm sure while sailing, some of you have dropped objects in the ocean. So if you were to drop a penny, I'm just curious, if you think about it, how long might it take for it to fall, say, 4 or 5 kilometers, the depth of the ocean? And if you think about it, for a penny, the answer is only a couple of hours. While if you were to drop a single cell, which can be hundreds of microns, it might take a couple years for it to reach the bottom of the ocean. And this is at the heart of the puzzle, that it's the fact that this sets of processes that capture carbon, the sinking velocities for much of whatever falls and dies in the ocean, that dictates how quickly we can capture carbon. And I think on the pressure variation, Deepak, I think the number is around 1 atmosphere for 10 meters, right?
That's right, yeah. Mm-hmm.
Yeah. Okay, so let's go back and think about how have we studied it in the past. One technique has been sonar. So many a times when you have all looked through in your boat to see and measure the topography of the bottom of the seafloor, an important aspect is what happened in the World War is the data that was collected for this reflection of these pulses going down and coming back up was the fact that, and this is again a story based around the first sets of submarines that were in the ocean, that they started calling this that the ocean floor is rising. And what they actually observed was the daily migration of life in the ocean. And I don't know if some of you have actually caught this while sailing, that there is a very high density of life in the ocean that's moving up and down, both in the morning and in the evening. And this vertical migration is what we're really interested in studying with the sets of tools that we will just describe. So let's just jump directly to the types of, these are the classes of tools that have been invented in the past. And as sailors, some of you might have actually seen these tools being deployed off of research vessels. Most of them are methods to collect what is sinking in certain kinds of traps. And what we are interested in is watching an organism fall and rise in the ocean as if you could attach a GoPro camera, for example, to that organism as it's sinking and rising. So we're going to talk a little bit about a new technology that we have invented that allow us to do that.
The history of this technology is really like what Manu said. Like, so if you wanted to study like a whale or a sea turtle, you know, like you could tag it and you could follow its movements using GPS. But how do you do that if the organism that you're talking about is microscopic? So it's like less than the width of a human hair. And so the idea is that you need to do microscopy. And what is interesting about microscopy is that like for the last 100 or so many 100 years that we have done microscopy, it's usually been restricted to the horizontal plane. So, you know, if you kind of look at a conventional microscope, this is kind of what it's going to look like. But for the purposes of studying things in the ocean, we're going to actually flip our perspective to look at microscopy in the other direction. So we're going to be looking along the vertical axis so that gravity can actually have an effect on the behaviors and the physiology that we are measuring. So this really led us to this idea of like, how would we do this in the lab where we let a single microorganism, which is say 100 microns in size. So that's like something that you cannot see. It's like a small speck of dust, and it's moving tens to hundreds of meters every single day in the way that Manu just mentioned. So the diel vertical migration in the oceans is actually one of the biggest components of the biological pump, along with the sinking of particles. So how do you actually measure a 100 micron organism that's moving tens to hundreds of meters? So to kind of give you a visual sense of scale, you know, that's kind of the, it's like as tall as a skyscraper. So that's the vertical scale that we are interested in. And, you know, the challenge with conventional approaches where, you know, you track a microorganism by moving your camera along with it, is that eventually you're going to hit a wall, right? And most of us don't actually own a skyscraper where we can track a microorganism that's going all the way 100 meters, which is probably like 50 stories. So what do you do to actually, you know, measure behavior over these vast vertical scales? And this is the question that we asked ourselves. And what we came up with was a very, very simple insight, but which was very profound and led to this idea of a hydrodynamic treadmill for single cells. So think about what you would do when you go on a treadmill. So you are running as fast as you can, as you would be running in an open field, but you're staying in pretty much the same place. So the same, you know, this is the spirit of the invention is exactly the same, where we actually have a vertical, we have essentially our endless column of water, which is, you know, approximated by this circular fluidic chamber. And what happens then is if you build a microscope at two special locations, either of these locations, if you can build a microscope, what you get is a hydrodynamic treadmill for single cells. And it works like this. So the organism is, you know, swimming through the fluid, but we are compensating for it by moving the fluid in the opposite direction by rotating the stage. So the way you should think of it is that this is actually vertically oriented, so like the face of a clock. And if you build a microscope at the 3 o'clock location, for example, and the organism is moving up, you can actually rotate the chamber in the other direction. And so even though the organism is swimming through the fluid, it's not moving in the lab reference frame. And what that means is I can do any kind of microscopy that I want and study plankton behaviors at a scale that's not been possible before.
Let's dive into, I think historically, something that's, in thinking about this idea, literally this idea originated in this office itself, and it's hard to see the whiteboard behind me. We sketched out this doodle a couple of years ago. And again, it takes a lot of work going from an idea to really implementing something. So I'm going to now transition into sharing with you very quickly, what does it look like in reality? And what can we do when we bring it to the ocean? And we will try, rather than going in detail, now we will spend a lot more time in just sharing with you, what does it mean to take a perspective of a cell? So you will be diving with us, and we will just be going through lots and lots of videos and movies to show you what does it feel like being a plankton in the ocean.
Mm-hmm.
So just to give you a physical perspective, this is what the machine looks like. We take this machine to several locations, including many of the marine stations around the world, but also to boats. The machine sits somewhere on a table, has a little wheel, and when it's operating, you can see the wheels turning. So literally, you can see right there, there is a wheel in the center that's turning, and this wheel is turning exactly equal to the amount that a particle is either sinking or rising. Let's watch a few organisms that you all love and care so deeply about, but in the context of this machine. At this point, we have, for the very first time, had the capacity to observe microorganisms as if we were swimming with them. Mm-hmm. So before this work, it was not possible to observe any of these microorganisms that you will now see in this level of detail from the frame of reference of those organisms themselves. So all the way from the left is an acorn worm larva. To many of you have probably recognized the sand dollars and the sand dollar larvae and the brittle star. And we're going to show you one or two examples of how surprising behaviors that these organisms depict. And much of this data is also now on the website called thegravitymachine.org that describes these.
What size are these microorganisms? How small are they compared to the
So all of them that we are showing today are less than 1 millimeter in size.
What about the needle? What about the head of a needle?
Yeah, I think smaller than a head of a needle.
Smaller than the head of a needle, yeah.
So this is Pteria miniata, or bat star, and I'll just show, just so that people remember, you've all seen this on tide pools in California. Mm-hmm. But of course, this grows to be an adult organism from a little larva that's only 100 to 500 micron. I think a speck of dust is a pretty nice analogy. If you think about it, that if you were to scoop some water, so if you're on a boat, scoop some water, you might find things that kind of look like, and again, they're called marine snow for this reason, is that many organisms and the debris in the ocean are just such tiny flakes, essentially. But when you observe them in a closest form, in a microscope, for example, so what you're doing here on the very left, you're seeing, you are apparently, the technique that we have developed allows you to apparently swim with the microorganism. So for the very first time, we have been able to swim with microorganisms. That is the crux of this technology. And then when you do that, you can watch the bizarre behaviors of these organisms that had never been seen before. So you're going to watch that in this video. On the very right is this little dot that we are tracking. This shows us the trajectory, and you can see the spiraling trajectory it's taking. You can compare it to the GPS track of your yacht, for example. But notice what the organism does now. So pay close attention to what's happening on the left. And right there, it took a dive, and what this microorganism is doing is it's feeding. And you can see what we have done is we have highlighted the flow fields of how the water is moving around the organism. And in the as detailed as possible, you are watching this behavior unfold right in front of your eyes. And this would have never been possible without a tool like this. Mm-hmm. And it's extremely important from a conservation perspective, is because now understanding the behavior of these larvae, we can start recapitulating how they are distributed. So when, for example, we had this big virus that was spreading all across our coast around, starting with Monterey and all across the Pacific coast, that was starting to kill many of these starfish, it was not clear whether these species would be able to come back and how these embryos would establish or reestablish themselves. And by studying these behavior, we get a chance to understand what types of decisions that these single larvae take in order to reestablish themselves in the environments that they care about. So after this, we're going to jump to one of the most exciting video that Deepak and I have ever collected using this gravity machine, and connect that back to the carbon cycle. So I'll let you walk through this one, Deepak.
Yeah, so I think one of the really amazing organisms that actually has a very large impact on our global carbon cycle are these phytoplankton called diatoms. And these are literally cells that live in glass houses. And this was us imaging this in a marine station in Puerto Rico. And what we saw really made us fall out of our chair. So this is a single cell that has no motile appendages. So it doesn't have any form of like swimming through the fluid. But what you see it doing is it's able to change its density over 100 millisecond time scale. So this would be as surprising if as someone told you, oh, I have a stone I'm going to give you, and that stone can decide whether to sink or float. So this single cell is actually now deciding to sink right there, as you just saw in this video, but very soon you're going to see it go back to just floating as though it's, like completely at the same density as a surrounding fluid. And, this is the kind of biological discovery that, makes us wake up every morning and come to the lab and makes us stay up at night, because, these are the kinds of organisms that are very important in understanding that vertical flux of carbon. And it looks like, we are only starting to scratch the surface in terms of their behaviors, which are directly related to sinking and therefore related to the carbon sequestration capacity.
All of this that you saw was data collected on land. And now we want to take you to, our research vessel that we've had the chance to spend some time on. And you'll appreciate why this is important, is because much of the biology that we can bring to the land is very different from biology in deep sea. So what you're looking at is a picture of Kilomoana. It is one of the longest running time series off the coast of Hawaii, where every 15 days to a month, this boat goes out into open waters. It stays there for 4 or 5 days and come back. And Deepak and I and Adam and a few students from the lab, we got a chance to take gravity machine on this boat. And what you're watching here is a video of us running the microscope on the boat itself on a cell that was caught just a few minutes before from the depths of the ocean, incorporating it in the wheel, collecting that data while there was also a storm coming. We were, a couple hours away from the edge of these high winds, and we were able to demonstrate that we can collect these types of data sets out at sea. And what's very important about doing this is, for the very first time, now comparing to the data set that you just saw Deepak sharing, in which the diatom was only sinking, here is another organism, a Halospharia, that is both rising and sinking depending on the time of the day we catch them. And it allows us to truly understand their native behavior as compared to the behavior of these sets of organisms in a lab setting. And to just show you a sense for, of course, for all of you, a rough weather is not that big a deal, but sometimes for us, when we are running these scientific instruments that can be a little more delicate, a few hours after that video was collected, this is what the ocean looked like. And of course, because we are on a boat, the entire microscope started wobbling, and we had to shut down data collection because the sea got too rough. But on the other hand, although this is difficult to do, it's extremely important for us to do this, because without these types of environmental conditions, we would never understand what it means to be able to understand much of this life that we have taken almost for granted most often, in what its native behavior is in the kinds of weathers. So at this point, we are starting to build many of these machines and deploy on boats around the world. And it's leading us to truly understand how to ask many of these very fundamental questions in biology, but in the context of the ecology of the ocean. If you want to learn more about this project and get engaged and involved and see all the videos that we just shared, you can go to this website. This is our team website called gravitymachine.org. And it describes at a very great detail all the instrumentation that went in, all the control systems and all the hardware for people to also be able to build these instruments. But most important, to understand and also watch all of the videos and the data that we shared. So I'm going to switch gears a little bit and for just 5 minutes describe a big project that we are just initiating that is primarily built around the sailing community. Up till now, we discussed the importance of studying these plankton, but the challenge that we face that we are only still a handful of scientists. If you think about all the scientists that are interested in the ocean, it is still a handful of scientists, but the ocean is a big place. And when I or anybody else who's studying the ocean wants to spend time in the ocean, I have to spend between $50,000 to $70,000 a day to rent many of these research vessels. This is an example of a research vessel out of UK. And so it's not sustainable for many scientists to spend a lot of time in the ocean because financially it's not possible. We started thinking about this challenge, and one of the biggest change in oceanography that has happened, at least in my lifetime, has been the introduction of sailing boats being recruited and used for science. The most famous of that is Tara. I assume some of you might have heard about Tara. This is an incredible boat that has literally changed the face of oceanography in the sense of the first sailing vessel that was fitted with scientific instruments to collect data sets. It's been on the cover of majority of scientific publications and the kind of data that it's collected. It's done expeditions that has been very difficult to do without this unique boat itself. And of course, the community that this boat has brought together in the scientific world. We started thinking about this, and again, this is in collaboration with Columban at Roscoff and a few sets of scientists across the world, to how do we recreate the success that Tara had as a sailing boat in the scientific world, but make that success accessible to everyone around the world. And that led to the beginning of the project that we call Plankton Planet. You can learn about this on the site called planktonplanet.org. And the insight was very simple. People just like you, between 10 to 15,000 sailors are around the ocean all the time. How could we use the capacity of citizen sailors to really bring science to people around the world, and especially to understand the complexity of the biology that's spread across the ocean waters? And the way we have decided to do that is to build scientific tools that we can provide as kits to any sailor that's interested. So this was the first project we started for 2 years. We called it a citizen science. And these are individuals all across in France, in US, and in New Zealand that we recruited. These was the first pilot program for 15 boats that sailed for 2 years, collecting plankton samples. So what is included in the kit that we provided the users is a little net, which you throw at the back of your boat. You collect some of the samples, and the samples that come in, you heat on any of the stove on your boat, and it forms this little cake. And this cake is what we use to sample and get genomic data out to understand and tell what was where when you are sailing around. We've been able to demonstrate that we can collect high quality data using these sailors that have never done any scientific expeditions before, all by themselves. And I think one thing that's very exciting to us in this perspective is to build a vision of doing this data collection where mariners, scientists, and artists and citizens are all collectively working together. And one tool that we are very excited about is to release a new microscope that is designed to operate on a sailboat. We call that Plankton scope, and I'm just going to briefly show you the picture of that instrument here. We first installed our first version of Plankton scope on a boat of Gurek, who's a very famous sailor from France. And one of the perspectives, this is the actual instruments. It's designed as a microscope. And what you do in it is you take some of that water that you just saw, you pour it in the microscope, and it will collect data automatically. So what you're watching, for example, is a video stream that if you are sitting on your boat and you have collected a wine glass of water from the ocean, you pour it in this microscope, and on your tablet, an iPad, or your iPhone, you would have these pictures of these microorganisms swimming by. And what we are doing with this is we are collecting essentially the abundance of microorganisms that were in that bottle of water. So if you look right here, this is a picture collected automatically of every single living organism that was in, say, that wine glass that you poured in, that you've just picked up from the ocean. And it gives you, for the first time, a realistic snapshot of diversity of life that you are sailing upon at the microscopic scale. And the goal in designing this project has been to the ease of use. So you don't have to learn to operate a microscope, or you don't have to have prior experience. All you do is you throw this little net outside your boat, you collect some water, you pour it in this microscope, and out comes this picture that, as scientists, helps us explain the diversity. And for you as a sailor, helps you appreciate the diversity of life on the planet. We have started sharing this tool with the community around the world. So there are many sailing communities. There are a significant number of communities in France and New Zealand. And one of the reasons I was very excited when Ron invited us to give this talk was to actually recruit and build a sailing community out of San Francisco that would be excited about taking these instruments on their boats. We are building the first 100 of these instruments, and we are looking for between 10 to 15 sailors who would be excited about joining the program. It could be an instrument that we can build, or a kit that could be rented at the yacht club before you go out. And what's valuable is by the time when you come back, you upload the data on the website, and it allows us to understand the complexity of life, but also how it's changing in our own environments, including the Bay, and essentially for some of you who go out further down. So with that note, I want to close this session. Much of what we mentioned can be found on two websites. One is called planktonscope.org, and the other one is called gravitymachine.org. And Ron, we can take questions and discuss any of the ideas that we just discussed.
Great. I have so many questions, just loads of them. They cover a broad range. First of all, the basic question, how does the ocean absorb carbon? Tell me the mechanism.
So the way you should think about this, that there is CO2 in our atmosphere, which is in a gaseous form. And because the ocean and the atmosphere has a very large interface, I mean, almost 70% of our planet's surface is the ocean surface. The gas that's in the atmosphere gets dissolved in water. Just like whenever you drink soda, you know that there is dissolved carbon. Very similar, in the ocean, there is dissolved carbon dioxide. Now, all of the life, for example, either photosynthetic or non-photosynthetic life, requires carbon to build every single cell, all the molecules that life is built out of. And it takes that CO2 and essentially takes energy, say that energy is coming from the sun if it's photosynthetic, and just like plants, makes biomass. Mm-hmm. So just like a plant grows on land, soaking in the carbon dioxide in the atmosphere and locks it in the content of its leaves and the tree trunks. And this is why we say that our rainforests are capturing carbon. Similarly, in the ocean, there is a floating forest. It's actually but on the other hand, the floating forest is made out of tiny, tiny cells
that you can't see.
that you don't see because they are so microscopic. And all of that, when they die, they sink to the bottom of the ocean. And if one of those cells sinks all the way to the bottom of the ocean, you have locked that carbon for 50,000 years. So from a technology perspective, if some of you are technologists, it is one of the most remarkable carbon capture technologies that has already existed and has been working full speed to save us. There is a calculation that shows if the biological pump, this process of fixing carbon, stopped tomorrow, our CO2 content will jump from 400 parts per million to 600 parts per million overnight, because just the dramatic flux that it is soaking in all the time, it would not have the capacity to, and that would be way above the tipping point.
And another way of thinking of it in the reverse is like, this pump is so effective that if it was not for, like, say, volcanic eruptions and more recently anthropogenic emissions, the carbon dioxide concentration in the atmosphere would actually drop to zero.
Because it's a suction.
If the pump stopped, if this organic natural process of microscopic animals falling to the bottom and taking with them chunks of carbon, if that stopped, what would happen tomorrow?
Our atmosphere CO2 numbers, 400 parts per million is the current CO2. It will jump to 600. That would imply overheating, of course, of the planet. But at that same time, every single thing that we associate with climate change linked with the CO2 rise will already cross the tipping point by orders of magnitude. Of course, there is said that we have a fixed carbon budget that we can release in the atmosphere that is only based on the fact that the ocean has been absorbing. And one of the challenges that we face is we don't understand this pump. So I cannot tell you if the temperature of the ocean increases by 1 degree, which has already been happening, how would it affect the functioning of the pump? If the salinity of the water changes, I cannot tell you. And again, it's a very complex pump because in the end, it's happening all around the oceans. So we need a better understanding of the ocean, independent of the fact that, of course, it's a remarkable food source and much of the life is tied to the ocean. If you really think about climate change, ocean has a very important role to play.
Okay, so Deepak, how does the density of life change in the water column? There's water in the top meter, and as you go down 100 meters, 1,000 meters, 3,000 meters, 10 miles down, I gotta use miles, remember. How does the amount of life per square inch or foot change? Logarithmically?
Yeah, so that's a very interesting question. And I think it's not possible to say it's the same everywhere in the ocean. But one general rule that you can keep in mind is that, photosynthetic life is at the base of the entire food chain. So all life in the ocean ultimately depends on these single-celled organisms that are fixing carbon through photosynthesis. And what that means is that these are going to be found mostly in the top 100 meters. I don't know what that is in inches, but that's where the photosynthetic life is found.
100 meters is a football field.
A football field, yeah. And what is interesting is that below that, there is this, what is called the mesopelagic. And the mesopelagic is kind of like this very mysterious place where we are still finding new organisms. Pretty much every single expedition that goes to the mesopelagic is going to find something new. There's like a pretty high chance of that. So that's where you have a lot of these deep sea fishes and things like that. So the biomass actually is, it's kind of size structured where the really small organisms are very much concentrated in the top. And as you go down, you see more larger organisms.
Really? So you're saying there are larger organisms as you go down? We're used to thinking about whales being at the surface. How much time?
That's of course correct. Yeah, so it's, as I said, it's not possible to just say that this is a universal trend in the ocean, but because photosynthetic organisms are all single celled and microscale and mostly in the microscale, they need to be in that top 100 meters to actually be able to survive.
And I think the challenging part of your question is we have not observed the ocean. We have not observed the ocean in the level of detail to truly be able to map. Whenever we do this kind of a vertical cross section of life all the way from the top to the bottom, it's done at a given point and a given time.
Mm-hmm.
And because of the dynamics of life, life moves up and down vertically, even in a single day. And at that same time, there are several regions of the ocean that are so understudied. The earliest expeditions that Darwin went on and were taken to the ocean were to answer this question, does life exist on the benthic? Where at the very bottom of the ocean, because people believe that if there was no light there, there might be nothing that exists. Now we know and understand that at the bottom floor, there is a tremendous amount of life, but all of it, or actually majority of it, receives nutrients from the top as things fall. So the things that we talked about, carbon capture, all of that organic carbon is then eaten by much of the life in the ocean at the bottom. And if this carbon flux, this rain of organic matter stopped, much of the benthic life would also actually vanish.
Great. So now, what's the distance that one of these microscopic organisms falls in your gravity machine, your wheel, where you're turning it constantly so that you can focus on one microorganism in the water column because you're moving the water around it? How far does it fall in a minute?
Oh, in a minute? Yeah, so it's actually
Depends.
in many cases, it totally depends on the type of organism.
They're slow.
So they're pretty slow, but you can imagine that in, like, for example, in many of these larvae, in like around like 10 minutes, you can cover half a meter.
10 minutes? So one of these larvae could fall a meter every 10 minutes.
When I say in this case, when it's larvae, they're actually swimming.
Okay.
So they're actually swimming down or swimming up.
So they swim, do they swim faster going down or going up?
So they typically swim faster going down because they're heavier. So it's harder to swim.
Helps. I think a rough number to think about this would be is keep 100 meters per day is a good number to think about in mind, even for a speck of dust. Nobody knows how far a single cell goes because
it could be in a neighborhood in the top 100 meters, or it could keep going. We don't know yet.
We don't know yet because this is the first time we have had a handle on a technology now that we can observe. And up till now, the longest tracks that we have made are only around a couple of hours, 5 to 6 hours. We're now stretching them to be able to watch a same cell for multiple days. But it would be fair to say that for the single cells, we actually don't know what that distribution looks like. And again, you know, the light after 150 meters, it's starting to get very low. So for a photosynthetic organism, it makes sense that it should turn around. But there are many organisms that don't care and will go deeper and then come back. Why
does it want to go down? I'm sorry.
Oh,
I wonder why the chicken crossed the road. In this case, why did the plankton want to go down?
I mean, so there are several reasons for that, Ron, but one of the things about the ocean is that, like, the light is higher at the top, but the nutrients are lower at the top. So the nutrient concentration goes higher as you go down.
And then
why is there more nutrients down below if there's more light up above?
For exactly the reason that things sink.
Because they accumulate.
So things just accumulate, falling, and there's more life down there, and a plankton takes years to fall if it starts falling.
Yeah.
Next, a quick question.
But it's an important thing. The other reason, and again, it's many theories around, the whole reason why plankton go up and down daily, there is also a predatory reason as well. That
what?
Predators, because zooplankton, who will eat many of these phytoplankton and things, they chase. And so you can think about it from a context of detection. So zooplankton, for example, which form a big portion of this biomass migration, they will rise in the night and then fall back in the day. And what they are doing is what is believed that they're trying to be harder to be detected, because when you go deeper, so if there is more light, it's better to go down so nothing from below can see you.
Got it. Right.
It's also
Kind of like
because,
staying in the dark, but when it's dark outside, you can come up because nothing from below can see you.
and many of these zooplankton have visual predators like fish and fish larvae. So it makes sense that you would want to kind of hide from your visual predators by
What do you
sinking
mean
downstream.
wishful? What do you mean wishful predator?
Like visual, like things
They
that
see.
they see you.
Oh, visual predator.
Mhm.
Yeah,
Mhm.
So what about whales? Don't whales eat plankton?
they do.
They do.
And they do, that's why they do these long lunges sometimes where they will catch this gigantic pool of krill, for example. And that's why, I mean, whales also do these long vertical scenarios where they will just plunge and take an entire column.
But now krill is so much bigger than plankton.
No, there are krills that can get smaller and smaller all the way to zooplankton size.
All the way to the size of a pin of a needle.
There is a lot of whales that will survive on organisms that are the size of a pin of a needle.
Really, and they have such filters in their
Exactly.
bodies so they can filter through and get the nutrients that they want.
Yeah.
So how long do plankton live? What's the life cycle like?
I think, again, nobody knows. It's a very specific, because there are millions of species. And one of the things to think about, they are all have been lumped in this category of plankton. Like, you know, often enough in science, there is something when you don't understand, you call it a soup. So plankton to most people is like a soup, while there is almost as much diversity of kinds of lifestyles as you would see in any other ecosystems as well.
If not more.
Yeah, if not more.
Yeah.
And one of the things here is, with the rise and sinking, you can think of it that one cell might divide once a day. So that's a very typical number to think about. And, you know, it's hard to say, oh, one cell turns into one mother cell and one daughter cell. So in some sense, you could think about that the lifetime of a plankton can be of the order of 24 to 48 hours.
Okay, so now let's talk about how sailors, yachtsmen around the world can help you. What size of a device would you put on their boats? And what would be their like daily activity? Is this 10 minutes a day to 2 hours a day? How much? So give us the size of the device. How much does it cost? And how long would you engage them? And how would they report back to you?
Yeah, so I'm just going to show you a few pictures of the kit itself.
Would they use a mobile app to communicate back to you? They connect the device to a mobile app?
Correct. So let me pull out a picture of the kit.
So while you're doing that, Deepak, so in other words, a yachtsman who wishes to help in this research would sign up planktonplanet.org on my screen right now. Okay. And then essentially they would then use this kit, which you're showing me, which looks like a net and a float and a cord and a couple of instruments. How long does it take to make your little biscuit, your scientific biscuit there? How do you do it?
Yeah. So I think the way to think about the logistics of this was version 1 of the kit that we had shipped for the first recruits of sailors. And now we are building a team of around 100 sailors around the world. This included a plankton net. It included a little suction pump, which essentially sucks all the water and dries on a normal aluminum foil, and you just dry it up in your cook stove and you put it in a Ziploc bag. You put a date on it and you keep it. When you arrive at shore, you just send these biscuits from the location of marking what GPS coordinate did you collect this water at. This was for the first version of the expedition that we did. Now for the second one of the feedbacks that we got from the sailors was they didn't like to slow down while they were sailing. And again, I can understand why. But this is also where science and communities have to work together. So we designed a new net, because these plankton net is a very fine mesh. So if you are really fast, you can rip the net. But on the other hand, a sailor might not want to slow down, and of course, especially in the open ocean. So we have now designed a new net, which is a new kind of a net. It's called a high-speed net, and it's very aerodynamic. This is the picture of the CAD for what it looks like. So this would be the second thing that you'll be able to throw without slowing down. And then the third instrument that will be in the kit will be what you see on the left side, this little brick-like object. This is where you do the imaging. So you take this water, and before making a little brick out of it, you pour it in and you essentially collect the imaging data. And in terms of the first sets of sailors that we would recruit, I think, the thing that we are trying to figure out is make the first 100 or so units and kits available to sailors who are committed to the science completely free of cost. So they don't have to pay for anything other than they share the data back to the community once they land. And then there is a second round of sailors, because we're also making all of these kits available more broadly for anybody who wants to engage. And so all of these parts are roughly $100 or so in parts. The net is roughly around $100. The instrument is roughly around $100. And then, of course, all the software and all the community website access is free.
Yeah, I think I was actually on the Derek Bayless off the coast of San Francisco, out beyond in the ocean outside the Golden Gate Bridge about 6 months ago. And people on the boat did collect plankton off the stern, the platform. And before this talk, I remember actually watching some of this citizen research take place. So Manu.
But the important aspect of this is to be able to standardize the tools, because one thing that happens is when people are using the same common tools, then we can compare the data back to back. So the same net and the same microscopes and the same sequencing techniques are important. So when a sailor from San Francisco collects this data, and a sailor from New Zealand collects this data, and a sailor from Philippines collects this data, they can all go into the same database and they can be compared one-to-one. So much of this is also about standardization, which is extremely important in oceanography, because we are trying to build these tools to record the ocean over a long period of time.
So Manu and Deepak, it's been a pleasure having you as guests on the Wednesday Yachting Luncheon. Manu, Prakash, and Deepak Krishnamurthy are bioengineering researchers at Stanford University, engaged in fascinating research on the smallest organisms in the biggest oceans. And thank you very much for being our guests on the Wednesday Yachting Luncheon. And with that, our luncheon is adjourned. Thank you.
Thanks so much for having us.